There we go. I don't. Uh,
Recording.
So I've learned that the reason I can't manage to start class on time, and I'll just tell you, I'm actually here about 5 minutes before. But I can't seem to get it to start immediately. And I'm not sure exactly what the problem is, but they can't seem to help me with Hack IT Tech. They don't really know what to tell me. Mother, don't help. Mother, don't help. She puts up with my frustration in the back here. First, any questions or concerns?
I have a question.
Go.
If because I'm like behind, so I'm wondering if I get all of my assignments turned in, do I still have a chance to get a passing
Yes.
grade?
Yeah, you do.
Oh, okay. I
But you
emailed
need-
you a little bit ago, but I just wanted to ask you too.
Did you email me this morning?
Yeah, it was pretty late. It was
Oh.
like 9 o'clock or a little bit after.
Okay, I can tell you that I'm scrambling to get myself ready in the morning, and I can
That's
tell you
what I
I don't
figured.
even look at them until, but yes, if you'll send me a list of what you need to do and when
Okay.
approximately you'll have it done,
Okay.
I will, I'll do it for you.
I can't remember what it was, but the one thing was locked. I'll make a list and then email it to you. But I
Yeah.
just wanted to ask to make sure it was still a possibility.
Yeah, that's a yes, that's definitely doable.
Okay, wonderful. Thank you.
You're welcome. On I sent out an email last night. Your first case study is posted. If you look right here, and if you look at your syllabus, you have 3 case studies. They're all worth 30 points each. The first one, if you look at it, it has to do with bones. It has to do with bones and osteoporosis and calcium. And so you should be able to answer the questions. You want to go back and look at the PowerPoint. Let me show you really quickly where it is. I was going to show you. So this is three different case studies, 30 points each. Let's see here. Bear with me just for a second. So right here at the very bottom, right underneath exams. When I click on it, and it will be due, and I'll try to repost this with a due date on it. It will be due. I think I had it so it was due November 4th. November 4th at midnight. But so it's talking about the lady who realizes that she has hypercalcemia. And if you remember when we talked about the bones, what that means that her calcium levels are too high.
Your email says it's due November 10th.
Is it November 10th?
Yeah, that's what the email says.
Okay, then November 10th is the right date. If that's what I said, that's what I said. So November 10th works. So what is calcium homeostasis? You guys, you know what homeostasis is. What is calcium homeostasis? Don't answer the questions out loud. Why is calcium homeostasis tightly regulated? Well, what happens if you have too much bone resorption? Too much bone being broken down and not enough being built up. What can happen if your calcium levels are too low? What can happen if your calcium levels are too high? If you go back and look at the end of the channel 7 PowerPoint, the answers are there. List 3 main hormones that regulate calcium homeostasis. So there's calcium citriol, calcitonin, and parathyroid hormone. What target organs do they affect and what are their actions? Using the information in table 1, draw 2 feedback loops that illustrate the physiological responses that act to restore calcium homeostasis. Explain how parathyroid hormone secretion is regulated. How does the body get vitamin D? Describe the pathways that produce it and its relevance to calcium homeostasis. The meaning, excuse me, Amelia understands she has high calcium levels, but she has, because she has too much parathyroid hormone. But she does not appear to understand where the calcium is coming from. Refer to your feedback loop and explain to Amelia the meaning of bone resorption and how excess parathyroid hormone affects bone resorption. How does the effects of excess parathyroid hormone on the kidneys and intestines contribute to high levels of calcium in the blood? Amelia wanted to avoid developing osteoporosis. What is osteoporosis and what is the mechanism that causes it? And I think I said there were 9 questions. I think there's only 8. Yeah, there's only 8 questions. You'll submit this in the Dropbox, excuse me, right here. What was I going to say? Give me 1 second to think. As my mom's banging around in the kitchen next to me. Shoot. Oh, when you post this and when you submit it, it needs to be submitted as a Word document, a JPEG, or PDF. If you submit it any other way, chances are I'm not going to be able to open it, and you can't get it. I can't grade something that I can't open. And so you need it can only be submitted in those 3 forms. If you don't know how to do that, or you don't think you have access to that software, you do have access to that software because you have access to Office 365 through your hack tuition. And you can do any of those 3 from Office 365. If you have questions about that, you need to call Hack IT Tech. And they will show you how to access that through Hack.
When is this due, Professor?
I think someone was just telling me, and I'll look it up right now. Someone was just telling me that it is due
The 10th.
the 10th. Bear with me just for a second. And of course, I'm looking at the wrong email. Give me 1 second here. Let's try it now. I'm guessing, Jamie, this is your email that I'm clicking on. Yeah, so November 10th at 11:59 p.m. All right.
Ask a question.
Oh,
I guess for everyone, I don't know, but I'm having like difficulties with the 8 and 9, just putting the right, like they have the skeleton skull, you have to put the words of to the picture.
right.
So has anyone used a specific chapter for that, or does anybody have any advice for me?
You can access the textbook, Lakeisha.
I did, but yes, I did, but it doesn't have the same exact photo, like picture. I
Well,
don't think.
you can access the PowerPoint because they're all on the PowerPoint.
Okay. Okay.
And they are all, if you go into your textbook, McGraw-Hill, different textbook. But if you access McGraw-Hill, bear with me just for a second here. I'm just going to click on one of these to go into the textbook. You access the textbook here. I'm going to come up to where I'm looking at the chapters. Move this over. Right. Here, the skeletal system, chapter 8. You're going to find more than you ever wanted to know about the skeleton and those anatomical features. Excuse me. All right, let's get started on today. I want to get through the majority of chapter 12 today. You're going to learn that as we move through these later chapters, we're going to move a lot faster. Chapter 2 is a long chapter, and I think it's more abstract and more complicated than probably any of the other chapters. And it takes a long time to move through it. But everything builds on those chapters at the same, a lot of things build on that chapter. And so I don't ever think it's a bad thing to spend some time with it. However, once we get through that chapter, we start moving a lot faster. And so I want to get through the majority of chapter 12 today. So fundamental properties of neurons. Neurons are excitable. They respond to environmental changes. They're conductive. They respond to stimuli by producing electrical signals that are quickly conducted to other cells. When the signal reaches the end of the nerve fiber, the neuron secretes a neurotransmitter that stimulates the next cell. So excitable responding receptors respond to a stimuli in the environment. They conduct an impulse, an electrical signal. When the signal reaches the end of the nerve fiber, the neuron secretes a neurotransmitter, which then stimulates the next cell. If you remember this from the other day, this is a reflex arc. You've seen this before. You've seen the feedback loops. It was chapter 1. If you remember, a feedback loop starts with a stimulus that's sensed by a receptor. Then there's a signal that moves to an integrating center or an interneuron. The interneuron makes a decision about how to respond, or if we're going to respond at all. It sends out an impulse along a line saying how to respond. An effector carries out that response. So physiology is all about feedback loops. There's two main types of nerve cells. There's over 1 trillion neurons in the nervous system. So to keep that in context, it's about 10 times more neurons than there are stars in our galaxy. The neurons branch so extensively that they make up to about 50% of the volume of the nervous system. These specialized cells transmit signals throughout the body. Neuroglia play a major role in support and nutrition of the brain, but they do not manipulate the information. There are about 10 to 50 times more neuroglia than there are neurons. So for about every neuron, there's at least 10 neuroglia. 10 to 50 more neuroglia. So you're looking at 10 to 50 trillion neuroglia in one person's nervous system. With regards to classes of neurons, functional classes of neurons, we have sensory or afferent neurons. Afferent means incoming. Okay. Trying to get rid of my little pop-up that just decided to show up. Remember when I type in keywords, remember these keywords, because a lot of times you will see them over and over and over again. Afferent means incoming or entering. Efferent means outgoing or exiting. So afferent neurons are sensory neurons, and they're specialized to be able to detect heat, light, pressures, chemicals, and transmit information about them to the central nervous system. It begins in almost every organ and ends at the central nervous system. So this neuron right here. So these neurons are part of the peripheral nervous system. Some of the receptors are sensory neurons themselves, and others are separate cells that communicate directly to the sensory neuron. Interneurons are confined solely to the central nervous system, and they make up about 90% of the neurons. They receive signals from many other neurons and carry out integrated functions of the nervous system. They process, store, and retrieve information and make decisions about how the body will respond to stimuli. Motor or efferent neurons send signals to effectors, the muscles and the gland cells. So these are outgoing or exiting neurons. So one thing that you want to remember here is sensory neurons are bringing in information to the central nervous system. Motor neurons are sending signals out from the central nervous system to effectors, which can be muscle or gland cells. Do you see the different parts of a feedback loop? So it changes sensed. Signal takes the fact that there's a fact that there is a change. The interneuron receives that signal. The interneuron decides what to do with that signal. Sends a message out via the motor neuron to the effectors that will carry out the response. When studying for an exam, you reach for a beverage. To extend your arm, your what nervous system is activated?
Motor.
Find one that's
Right,
on this
parasympath-
list, but you're close.
Parasympathetic.
Nope, nope. Parasympathetic is autonomic, so involuntary.
Sympathetic.
That's involuntary too. Go back and look at right here. So it carries signals to the skeletal muscles. So it's a somatic nervous system. That's your voluntary nervous system. Because if you reach for a beverage, you made a voluntary movement towards that beverage. Is that clear? An example of a neural feedback loop. So sensory function. You smell the coffee, stimulates sensory receptors in the nose. Olfactory information is relayed into the brain where it's processed and the decision to drink the coffee is made. The brain stimulates the contraction of specific muscles that allow you to grab the cup of coffee and drink it. Any questions? Looking at the structure of a neuron, the soma is a cell body or the control center of the cell. It contains the nucleolus. If you remember, the nucleolus is where the rRNA, the ribosomal RNA, and the ribosomal proteins and subunits are assembled. It also contains other organelles, including the rough ER, that contains what are called chromatic Phillips substances or Nisi bodies, these little dots, and a cytoskeleton that includes neurofilaments. There are no centrioles here. What were centrioles involved in? Do you remember what's their function? Okay, I'm going to start picking some names. Miranda, what's the function of the centrioles?
I keep confusing it with something else. I thought it was to bring nutrients to the neuron, but if they're containing no centrioles, that would make no sense.
Right. Can you Google it really quick?
You want me to Google what are centrioles?
Yep. What are their function?
They are paired barrel-shaped organelles located in the cytoplasm of animal cells near the nuclear envelope. Centrioles play a role in organizing microtubules that serve as a cell skeletal system. They help determine the location of the nucleus and other organelles within the cell.
Okay, let me ask, so what are microtubules involved in? They're part of the cytoskeleton. It doesn't really give you the information I wanted it to give you, but they are involved in the separation of the chromosomes during mitosis. So if neurons don't have centrioles, what are they not able to do?
Not able to like regenerate.
Yeah, they don't regenerate, they don't divide. So keep that in mind as we go forward here. They're not capable of regenerating. If they don't have centrioles, they cannot divide. if you think about, actually, let me stop for 1 second here. What would be the implications of that? Fact that they don't divide.
If they're
Can you
damaged, and that's it.
Yeah, if something happens to them, you're kind of out. You're out of the game. So that is why nervous system injuries are so devastating. Because almost every other cell in the body may reproduce slowly, but it does reproduce itself and can heal itself. And the nervous system, more than not, will never do that. Dendrites branch off from the soma. These are little feelers. I like to think of them as little feelers that bring information into the soma, the cell body. So these are the primary sites for receiving signals from other neurons. The more dendrites a neuron has, the more information it can receive and incorporate into decision making. So the more branches there are, the quicker information is received. And the more information is received. And then the cell can decide what it's going to do. And we'll talk about how it makes that decision. Neurons can develop more dendrites the more they're stimulated. So the more often you stimulate a neuron, it will respond in kind and develop more dendrites. Specifically with your special senses, your hearing and your sight, your touch. It responds more in kind. At the bottom of the soma, or at the other end of the soma from the dendrites, there's an axon hillock. This is where the axon is going to originate. The axons are relatively unbranched, but axon collaterals can originate near the soma. Axons are specialized for rapid conduction of nerve signals. The cytoplasm is axoplasm, and the membrane is axolemma. So axo always means axon. Some words that are going to be good to remember. Plasm is always going to refer to a cell's cytoplasm, whatever the cell is. So the axoplasm is the cytoplasm of the axon. Cellplasm, cytoplasm. Or cytosol. Oh, what was I saying? What was I thinking? Give me one moment to think. Lemma is the plasma membrane. Anytime you see those words, that's what they mean. these axons may be myelinated, and we'll talk about what myelin is here in a moment. The neurolemma is the most outer edge of the myelin. Most outer edge of the myelin. At the distal end of an axon, there is usually a terminal arborization. Each branch ends in an axon terminal that forms a synapse with the next cell. What does arbor mean? Does the word arbor mean? A-R-B-O-R. Or have you heard that word before?
Like a tree?
Like a tree, yes. So if you look at the end, what it is it's saying that the end of the axon ends in a branching or a trunk of branches. So each branch ends in an axon terminal. Each one of these is these little tiny branches, little almost like looks like dendrites, but on the axon instead, are going to terminate on and synapse with another cell. Think of synapse as meaning a snap, where one thing joins up with another thing. So each of these branches is going to synapse with the next cell. Each axon terminal contains synaptic vesicles containing neurotransmitter. So each one of these contains vesicles that contain neurotransmitter. If you want to think of this, here's your tree. Here's the branches of the tree. The axon is the trunk. And the terminal arborization is the root system. But rather than have things move from the roots to the leaves, like in a normal tree, it moves in the reverse here. So the terminal end of the axon has this extensive branch of complex of fine branches. That each end is a synaptic knob or a terminal bouton, where there's a swelling that's going to form a junction with the next cell. The neurotransmitters are signals used at synapses to pass an excitatory or inhibitory signal from a neuron to its target cell. So these neurotransmitters that are released can either work to excite another signal in the neuron that it synapses with, or the cell that it synapses with, or it can work to block a signal from occurring, block that next cell from firing. Any questions so far?
No.
So there's three structural classes of neurons. There's multipolar neurons that have one axon with multiple dendrites. This is the most common type of neuron. And it is most of the neurons that are found in the brain and the spinal cord. Bipolar neurons have one axon and one dendrite, just like if you've ever, I have nothing against people who are bipolar. I have many friends who are bipolar. I have family members who are bipolar. But what does that mean? It means that oftentimes their personality can swing from one end to the opposite end. So bipolar neurons have one axon and one dendrite. These are specific to the special senses. You only find these in the special senses. They're found in the olfactory cells in the nose, certain neurons in the retina of the eye, and sensory neurons in the ear. Anaxonic neurons have multiple dendrites and no axon. They communicate locally through their dendrites but produce no action potentials. They're found in the brain, the retina, and the adrenal medulla. So unipolar neurons have a single process leading from the soma. This would be our, the other word for these would be our bipolar neurons. Bipolar neurons have a single process leading from the soma, carry signals for senses, touch. Actually, I was wrong. These are not bipolar neurons. Unipolar neurons have a single process leading from the soma. They carry signals to the spinal cord for senses such as touch and pain. They have single processes that stem off with a peripheral fiber and a central fiber, with the cell body in the center. These are specifically your sensory neurons, your afferent neurons. Your sensory afferent neurons. We'll see this when we talk about the spinal cord. So sensory afferent neurons. So the single process stems off into the peripheral fiber and the central fiber. Peripheral fiber has a sensory ending far from the soma. The signal travels along the peripheral fiber to the central fiber and then to the spinal cord. This is a two-way passage of proteins, organelles, and other microtubules along the axon using microtubule monorails via axonal transport. There's two different motor engines that function along these rails, and it depends on what direction we're transporting materials. If material is transported towards the axon away from the soma, we call it anterior grade transport. And a motor engine called kinesin is used. If things are moving towards the soma, towards the soma away from the axon terminal, it's called retrograde transport. And the motor engine is dyning. So these are if you remember, microtubules are cytoskeleton, part of the cytoskeleton, and these are essentially the roads that allow for things to be transported down the axon or back towards the cell body. fast axonal transport occurs at a rate of about 200 to 400 millimeters a day. We can have fast anterior grade transport. It's going to move mitochondria, synaptic vesicles, and components of the axolemma, calcium and enzymes toward the distal end of the axon. And we can have fast, or we can have fast retrograde, which is moving things back towards the cell body, informing this and allowing for the soma, the cell body, to be informed of conditions of the axon terminal. This retrograde transport is often taken advantage of by pathogens such as the tetanus toxin, rabies, and the poliovirus that take advantage of this process to reach the cell body of the neurons. Wonder if this is also how chickenpox works, but I'm not sure. Questions? Slow axonal transport works only in an anterior grade process and is a stop and go process. So we have fast, which happens in both directions, moving towards the axon terminal or moving back toward the cell body. Occurs at a rate of 200 to 400 millimeters a day. We have slow, which is stop and go. This is kind of like, this is meaning the direct route. This meaning that taking the bus that stops at every stop. Or the plane that has frequent layovers. It moves at about 0.2 to 0.5 millimeters per day. This is how neurons will move enzymes and cytoskeletons down the axon, renew components, and supply new axioplasm, new cytoplasm for developing and regenerating neurons. We'll talk about that regeneration. If the soma is not injured, a lot of times things can regenerate to a certain level. They can't divide, but they'll regenerate. Neural glia are not electrically excitable. Glial cells function to serve, protect, and support neurons. If you look here of the central nervous system, the astrocytes are a neuroglial cell, ependymal cells, microglia, and oligodendrocytes are all neuroglia of the central nervous system. The oligodendrocytes have a bulbous body and up to 15 arms. I think of them as almost looking kind of like spiders. And one body and lots of legs or lots of arms. Each arm reaches one axon terminal and spirals around it. So this wrapping is the myelin sheath that insulates the nerve axons and speeds up signal conduction. What this means is that if it has 15 arms and one arm reaches for an axon terminal and spirals around it, how many neurons can one oligodendrocyte possibly assist in myelinating.
Up to 15?
Up to 15, right. We're going to see it's very different in the peripheral nervous system. It doesn't work that way there. But up to 15. Some of those arms, depending on what the oligodendrocytes location and with regards to other neurons, some of those arms may form the myelin sheath around the same neuron. But it can do up to 15. Ependymal cells resemble the cuboidal epithelium and it lines the cavities of the brain and the spinal cord. However, it's not cuboidal epithelium in that it does not have a basement membrane. It secretes a cerebrospinal fluid that bathes the central nervous system and fills the cavities. Has patches of cilia that help the cerebrospinal fluid to circulate. And little cilia, cuboidal epithelium. Microglia are small macrophages. What does phage mean?
To eat.
To eat, yes. So this is a cell that a bit macro meaning big cell, big. So this is a big cell that eats other cells or eats other things. Don't bark. My mom's banging around in the kitchen and my dog here on my lap is thinking about barking. Don't bark. So microglia are macrophages that develop from monocytes, which are cells that perform phagocytosis and parts of the immune system. They wander through the central nervous system, putting out finger-like extensions to look for cellular debris or other problems. They phagocytize dead tissue, microorganisms, and other foreign material. They're concentrated in regions that are damaged by infection, trauma, and stroke. They aid in the repair, the synaptic remodeling, and changing of connections between neurons. Constantly, as you're sitting here learning about physiology, your brain is undergoing synaptic remodeling. Astrocytes, astro meaning starlight. So these are many branched starlight shaped cells. They constitute over 90% of the cells in the areas of the brain and cover the entire surface of the brain and most non-synaptic regions in the gray matter. Astrocytes have the most functions of all the central nervous system neuroglia. And we're learning more and more about all that they do. But to keep things fair, reasonably or within limits simple, we're just going to talk about 9 of the functions that they have that are well known. So astrocytes provide the supportive framework for the nervous system. They have perivascular feet. Peri meaning around, vascular meaning blood vessels. So they have perivascular feet that contact the blood capillaries and stimulate the formation of the blood-brain barrier. They monitor neuronal activity and stimulate dilation and constriction of blood vessels to regulate and constrict blood vessels for blood flow to alter blood flow for changing needs. They convert the blood glucose levels to lactate and supply this to nourish the neurons. So all the glucose has to be converted to lactate in order for the neurons to be able to use it. And this is done by the neuroglia. So blood-brain barrier, supportive framework, stimulating and regulating blood flow to meet changing needs, and converting blood glucose to lactic acid to supply the neurons. Moreover, they secrete nerve growth factors that regulate nerve development. They promote synapse formation and fine-tune circuitry. So they're also involved in neuronal remodeling. They communicate electrically with neurons and can influence the signaling patterns of the synapses. They can regulate the tissue fluid absorption through absorbing released neurotransmitters and keeping potassium from reaching excessive levels. We'll talk about how before the end of today, I hope, we will talk about how potassium is vital for nervous system signaling. Astrocytes also perform sclerosis or astrocytosis, where they fill in space that was formerly occupied with damaged neurons with scar tissue. We've heard of multiple sclerosis. Those are scar plaques that are formed. Any questions? Which of the following is not a function of astrocytes? Astrocytes control the chemical environment around the neurons. They anchor neurons to blood vessels and provide the blood-brain barrier, provide the immunological defense for the central nervous system, or absorb and recycle neurotransmitters.
C.
C. You're right, it's C. Provides immunological defense for the central nervous system because that's the microglia. In the peripheral nervous system, we only have two types of neuroglia. We have Schwann cells or neural lemocytes. Remember lemo meaning, this is neural lemma meaning the outer layer of the myelin around the axons. So neural lemocytes would be the cells that form the myelin. So these Schwann cells wrap repeatedly around the nerve fibers of the peripheral nervous system, producing a myelin sheath. So these have the same role that the oligodendrocytes have in the central nervous system. They're a little different, but they have the same role. Satellite cells surround the soma in the ganglia of the peripheral nervous system. Ganglia are what contain the cell bodies. They provide the insulation around the soma and regulate the chemical environments of the neurons. Gliomas in the central nervous system and peripheral nervous system. Most neurons rarely undergo mitosis, so they seldom form tumors. Therefore, tumors are going to have to arise from the mitotically active glial cells. They grow rapidly and are highly malignant. They're resistant to chemotherapy. They're resistant to chemotherapy because of the blood-brain barrier. This person right here, this is actually a picture of my cousin. He had a brain tumor removed about a little over a year ago. And he's actually doing really well. He just ran the St. George Marathon about a couple weeks ago. So he's doing really well at this point. But understand why we don't see tumors of neurons. We don't see them because they don't undergo mitosis. And if cells are not going to divide, we're not going to develop tumors from them. And because most chemotherapy is given through the bloodstream, brain tumors are going to be resistant to chemotherapy because of the blood-brain barrier. Questions? You guys, I don't want to take the full time. Let's take 7 minutes. Come back here at 10:45. What'd you say? She says, look at my face. Do you think I could ever do something like that? find her primary orange buckets. Space. Still love our apartment. Still love that. So the myelin sheath, oh, and I forgot to grab something. This side. Oh, that was great. One moment. Okay. So the myelin sheath is a spiral layer of insulation around the nerve fibers that's formed by the oligodendrocytes in the central nervous system and the Schwann cells in the peripheral nervous system. This insulation works similarly to the insulation around your extension cords or your electrical wiring. What does the insulation do around your electrical wiring in your house? What does it do? Aaron, what does it do?
The insulation on wiring in the house
Yep.
keeps other things from interacting with it. It keeps the signals, the electricity inside where it's supposed to be, keeps it focused.
Yep, it keeps it focused. It speeds it up. If you think of it that way, it speeds it up. So it keeps it focused, keeps it from leaking out. And that's what the myelin is doing here. So the myelin has the composition of the plasma membrane with about 20% protein and 80% lipid. So I have the way to picture the myelin sheath. Oh, 1 second here. I have this, here's my axon. And I have a cell, and I just keep wrapping the cell around, wrap it really tightly around part of the axon. Wrap it around really, really tightly. What else is in a cell besides the plasma membrane? What else makes up a cell? What else makes up a cell?
The nucleus makes up a cell.
The nucleus makes up a cell. What else, Quentin?
Mitochondria, endoplasmic reticulum.
Yep. So what happens if I keep, where does all that go? If I keep wrapping solely the lipid bilayer around this part of the axon, where do all those other organelles go? They get squished to the outside. And so the majority of what actually is doing the insulation here or almost all of what's doing the insulation here is the plasma membrane. With everything else being squeezed out. You look here, here's a cross section through one. Here's the nucleus out here. So the composition of the myelin sheath is like that of the plasma membrane. The production of the myelin sheath starts around week 14th of embryonic development. However, there's very little myelin in the brain at the time of birth. Means that things are kind of slow. Myelin is also very important in the formation of the synapses. So when they say that babies are learning a lot really fast, and they're learning more than you recognize, they are, because that myelination is occurring rapidly during infancy, because that's very much involved in the development of the nervous system. This is also why they tell you not to put babies on low-fat milk and give them only whole milk until they're about 2 years of age. Is because the dietary fat's so important. So the Schwann cells in the peripheral nervous system can lay up down to about 100 compact layers of the plasma membrane, with that most outer coil being the neurolemma. Then there's a basal lamina, remember the basement membrane, right? Basal lamina. And then a coating of fibrous connective tissue, the endoneurium. In the case of the peripheral nervous system, it's one cell, one segment. One cell, one segment of myelin. One cell, one segment. One oligodendrocyte in the central nervous system can myelinate several nerve fibers. If it's one cell in the myelin per segment of the myelin sheath in the peripheral nervous system, those Schwann cells can move wherever that axon moves. Ever one because it's one cell per one segment of myelin. Oligodendrocytes, because they can work on their different little arms, can be involved in multiple segments on multiple nerve fibers. They're anchored in place because they're attached to multiple fibers. Schwann cells wrap from the inside out. So they start at the inside and they wrap and wrap and wrap and wrap and wrap and wrap and wrap. oligodendrocytes actually move newer layers underneath older layers. So the myelination in the peripheral nervous system myelinates from the spirals outward. Myelination in the central nervous system is going to spiral inward. So in the case of the central nervous system, there is no neural lemma, meaning the outer layer. There is no neural lemma. There is no basal lamina, because newer layers are being put in underneath older layers. And there is no endoneurium. That's significant later. That matters. Questions? Many Schwann cells or oligodendrocytes are needed to cover one nerve fiber. Since the fiber is much longer, the axon, the nerve fiber means axon. Nerve fiber. It's axon. In the peripheral nervous system. Mom, can you turn it down a little bit? Yeah. We will talk about this here in a minute, but nerve tracts. Actually, this is groups of axons. Oh, wait, no, we'll leave it that way. Nerve tracts are groups of axons in the central nervous system. So many Schwann cells and oligodendrocytes are needed to cover one nerve fiber, since one fiber or one axon is much longer than the reach of a given glial cell. Therefore, the myelin sheath is segmented. So where one cell ends in its myelin sheath coating, another cell will meet. The gaps in between the different myelin sheath parts are nodes of Ranvier or internodal segments. So just to reiterate, here's our axon. Here's the axoplasm, which is in the axon. Axolemma is the plasma membrane around the axon. In the peripheral nervous system, the Schwann cell makes up the myelin. The most outer layer is the neural lemma, plasma membrane. Moving back a little bit and talking a little bit more about the axon. The axon hillock right here, and the initial segment of the axon is called the trigger zone. This is important in initiating a nerve signal. We'll probably talk about this before the end of today, but how that is initiated. But this trigger zone is very important for initiating a nerve signal. It's going to go down the axon. Multiple sclerosis is an autoimmune disorder where the myelin sheath of the central nervous system is attacked by the immune system. Autoimmune means the body is attacking itself. And I'm pretty sure we haven't talked about this before. Autoimmune. Meaning the immune system, one's own immune system is attacking itself. As you go forward with your physiology studies, and as we move forward, you're going to learn that a lot of disorders are caused by one's own immune system attacking itself. I think auto is meaning oneself. So it may be triggered by a virus in genetically susceptible people, maybe more susceptible to getting infected by a virus that initiates this autoimmune attack against one's own myelin in their central nervous system. So the attack is essentially against the oligodendrocytes and the myelin sheaths, where the sheaths are deteriorate and are replaced by hardened scar tissue. Do you remember what neuroglia puts down the scar tissue? Yeah, the choice of 4. What one was it? Microglia, wasn't the microglia. Ependymal cells. Microglia are in the immune system, but they're not involved in the scar tissue put down. Ependymal cells, what do? Catherine, what do?
Oh, I'm not sure.
They're going to secrete the cerebrospinal fluid. What other ones do we have? We have oligodendrocytes, but they're the ones being attacked here. So what's the last one that we have? I think it's Ashley. Ashley Thompson, are you here? Or Asante? I think it's Asante. Maybe. I may be really wrong because it just says iPhone 2. Miranda, what is it?
Oh, that's me, Michaela. I don't know. I'm just like listening.
I want you to be actively involved. I try to keep you actively involved, trying. What,
Yeah, I have no clue with this one.
it's the astrocytes.
Okay.
Okay. I'm going to keep you involved. I'm trying to keep you guys actively learning. I know it's not always easy, and I know if this class is early for you, it's early for me.
Mhm.
But I'm trying to keep you actively involved. What these different symptoms, they can be variable. Many people die within a year. Some people die within a year of diagnosis, but others can live for 20 to 30 years. So it's just like having the myelin break down around, having if you think of it, having the insulation of the wiring around your house, maybe having a short in the wiring. All of a sudden things slow
Okay.
down a little bit. It's a little harder to get the same thing done. Each neuron in our body has a span of how long?
Is it D because they don't reproduce?
Yep, it's D, an average lifespan. Good job, Erin. Unmyelinated nerve fibers. Many nerve fibers in the central nervous system and peripheral nervous system are not myelinated. They are, rather than having several layers of myelin wrapped around them, they are enveloped in Schwann cells. So what in the neural lemma, the outer layer, that neural lemma, they kind of move into the folds of the myelin sheath of other axons, but they're just in the most outer layer. So one Schwann cell can harbor 1 to 12 nerve fibers within the grooves. And the fold just wraps once around each fiber. So in the outer layer of the myelin. Some terminology. Neuronal cell bodies are often grouped together in clusters. Remember, these are the somas. Ganglions are clusters of neuronal cell bodies in the peripheral nervous system. The nucleus is a cluster of neuronal cell bodies in the central nervous system. You guys, over the next couple weeks, definitely learn these terms, these 6 terms, because we're going to talk about it a lot. So ganglions are clusters of cell bodies in the peripheral nervous system. The nucleus is a cluster of cell bodies in the central nervous system. In the peripheral nervous system, a bundle of axons is a nerve. In the central nervous system, a bundle of axons is a tract. The nervous system is also grouped as regions of gray matter and white matter. White matter is highly myelinated. Gray matter is not so highly myelinated. It doesn't have any myelin. So white matter is the aggregation of myelinated axons of many neurons. The lipid part, remember how it's mainly lipid. So the lipid part of the myelin gives the white appearance. The gray matter is formed from neuronal cell bodies, dendrites, and unmyelinated axons, and it lacks myelin. So this is cell bodies, dendrites, and unmyelinated axons and neuroglia. This is myelinated axons. Nerves are found in the what, while tracts are found in the what?
Would it be A?
It's A. So nerves are found, Isabel, that's correct. Nerves are found in the peripheral nervous system. Remember, what are they? Isabel, what are they? What are nerves? How do you define a nerve?
The bundle of axons.
And they're myelinated axons in the peripheral, bundles of myelinated axons in the peripheral nervous system. Tracts are bundles of myelinated neurons in the central nervous system. So the conduction in axons depends on two factors: the diameter of the fiber. So the signal conduction is going to occur along the surface of the fiber. So a fiber with more surface area is going to conduct signals faster. And the presence or absence of myelin. In smaller unmyelinated nerve signals, the signals travel about 0.2 to 0.5 millimeters per second. In the myelinated fibers of the same small size, nerve signals can travel 3 to 15 meters per second. In large myelinated fibers, the nerve signals can travel up to 120 meters per second. So why don't we want all of our nerve fibers to be fast? Why aren't they all myelinated and large diameter? Our brain would probably blow up if it received all the signals really, really fast, I would think. It's probably what my mother's laughing in the other room, but they would probably blow up. Maybe not, but it wouldn't be a good thing. We need some to function a little slower, like secreting stomach acid or dilation of the eyes pupil. We need things to make some things to move a little slower while some things move a little faster. Fast myelinated fibers function in processes such as motor commands and sensory signals for vision and balance. We need some things to happen really, really fast, but we want some things to happen a little slower. Tay-Sachs disease is a hereditary disorder where there's an accumulation of a glycolipid in the myelin sheath. Normally, what it is there is, do you remember where lipids are broken down within the cell? Where are lipids broken down within the cell? Somebody tell me. If you want to, look it up.
Capillaries.
They're not in the capillaries, but you're close. What about the peroxisomes? Or the lysosomes? So the glycolipids are broken down normally within the cell. so they never accumulate to large amounts. However, when there's an enzyme that's not being produced that's supposed to break down these glycolipids, when that enzyme is defective or not produced, those glycolipids are just going to accumulate. What do you know about structure and function? Well, if you impact structure, you're going to impact function. If there's a swelling, because there's so much glycolipid accumulating, eventually that's going to affect function. So the glycolipid accumulates because there's no way to break it down in the myelin sheath. And this ultimately is going to disrupt nerve conduction. If I remember right, children with Tay-Sachs disease usually live 2 to 3, maximum 2 to 3 years after diagnosis. And the symptoms that come with it are blindness, loss of coordination, and dementia. If you look here, this is a cross-section through a nervous system tracts. So death usually occurs after at least within 2 to 3 years of diagnosis. Any questions? A damaged peripheral nervous system may regenerate. So some neurons can regenerate. That doesn't mean they divide. It just means that they can regenerate. So a damaged peripheral neuron, if I remember, this is the peripheral nervous system, can regenerate if the soma is intact. What's the soma? Soma is a cell body. And some neural lemma. What was the neural lemma? It's a neural lemma. Most outer layer of the myelin sheath. If the soma is intact and some neural lemma remains. If the fiber, so this is the process by which things can regenerate. At the time that the nerve is severed, the fiber that's distal to the cut will not survive because it is incapable of synthesizing proteins, because that occurs at the soma. So anything distal to the cut is not going to survive. So the Schwann cells degenerate, and the macrophages, remember, these would be glia, or in the peripheral nervous system, other immune cells that will clean things up, clean up the cellular debris. The soma loses the effects of nerve growth fibers from the neuron's target cells. Down here, the target cells. For this reason, the soma will swell up, the cell body will swell up, the endoplasmic reticulum breaks up, and the nucleus moves off center. Many neurons die at this point. So some of the way that neurons survive and function is based on nerve growth factors that are coming from the target cells. And therefore, when there's a severing of the nerve fiber, this it can cause damage to the cell body. At this point, if the cell survives, if the neurons can survive, the axon stump will start to put out growth processes. When cells lose their nerve fibers, they can undergo denervation atrophy, meaning they shrink and they shrivel. However, if possible, if the nerve can recover, it will we'll see that it can reach out again to its targets, the nerve muscle cells. But so the axon stump sprouts growth processes. Near the cut, the Schwann cells and basal lamina and neural lemma will try to form a regeneration tube. So what the Schwann cells, the basal lamina and the neural lemma, what are they part of? They're all part of the myelin sheath. So distal to the cut, the Schwann cells, the basal lamina, and the neural lemma will try to form a regeneration tube. When the growth processes from the zoma that came out, you remember that the axon stump can sprout these growth processes. When the growth processes enter the regeneration tube, it can grow rapidly, while other growth processes retract. Multiple growth processes will be put out. If one of them can enter the regeneration tube that forms from the myelin sheath, distal to the cut, the other growth processes will then degenerate. And the growth process that entered the regeneration tube will guide that recovering neuron back to the original target cells. And synaptic contact will be reestablished. When the synaptic contact is reestablished, the soma can then receive the nerve growth factors that come from the target cells. So the soma will recover and shrink back and return to its original appearance. And the reinnervated growth fibers will regrow. Questions? So Schwann cells in the endoneurium are required for nerve fiber regeneration. Because the central nervous system lacks Schwann cells in the endoneurium, central nervous system nerve fibers will not regenerate. However, because the central nervous system is enclosed in a lot of bone, it tends to not get damaged as easily. I can't imagine that you guys don't have any questions.
How long does that regeneration process take?
It can take a really, really, really long time. It can sometimes take several years. It may take less than that. I think the last time I really had a nerve damaged, at one point when I was living in St. George, Utah, I went to see the dentist, and when he put the anesthesia in my mouth, when he injected me with a needle in my mouth, I know he hit a nerve. And let me tell you, that hurt. But I think that was, I don't know if he really severed the nerve. He obviously poked something. And I was numb there for probably a couple of weeks. When I broke my jaw, they had to sever a bunch of nerves to put my chin, to fix some of my chin back. And they knew at that point that some of those nerves may or may never, that their comment was they may never regenerate. They may never come back to complete feeling. I think they did. I can feel my chin. It depends. So that's a really good question, and it doesn't really have a clear answer either. How does a neuron generate an electrical signal? And how does it transmit a meaningful message to the next cell? If you remember when we talked about the cell membranes, we talked about electric potentials. This is a difference in the concentration of charged particles across one point and another. You guys, what's a potential energy source? What's a potential energy source? A potential energy source is an energy source that can
Stored
be used.
energy. Oh.
What? Say it again, Miranda.
I was gonna ask if ATP would be a potential
It
energy.
is. It is in its way. It's not a very stable one, but it is one. It's not very stable in that we just make ATP so we can use it and we can't store it. But glucose is one. A concentration gradient, because remember how things like to flow downhill. Nothing likes to be highly concentrated, especially a bunch of light charges. A bunch of positive charges together don't like to stay together. If we allow them to flow down a hill, that is a potential energy source. Once we let them move, that's a kinetic. So when they want to move, but we don't let them, that's a potential energy source. When we let them move down their concentration gradient, that's now kinetic. So anytime that there's a stored concentration, that's a potential energy source. ATP, like Miranda said, that's in its form of the adenosine and the ribose sugar and the 3-phosphates. That's a kinetic, that's a potential energy source. Glucose is a potential energy source. In the case of electrophysiology or neural communication, anytime that there's a stored concentration gradient, that's a potential energy source. A current is where we allow those charged particles to flow. Just like your electrical currents of your wiring at home. I'm gonna do something really quick. Here I have oh, we don't want that big of a battery. So here I have a battery. Okay. On one side of the battery, this is my typical 9-volt battery. So this is my Duracell battery that I have at home. On one side of the battery, there's charges all concentrated together. On the other side of the battery, there's charges all concentrated together. Just like that. Why does this battery help me to have it at home? Why do you keep batteries in your house?
Remotes, fire, alarms, things like that.
And how does it do that?
The electrical charge, they like contradict each other. There's a
They contradict each other. They do a little bit. They do a little bit.
complement's a better word, I believe.
They complement one another. There is a separation in charge. Have you ever licked a battery?
Yeah, it zaps your tongue a little bit.
Yeah, why does it do that?
I have not a clue.
Because it creates a current where one side of the charges from one side of the battery flow to the other side of the battery. So the negative charges are attracted to the positive charges on the other side of the battery. And so when we create a current here through our saliva, the negative charges will then flow over to the positive charges. You guys, if you never licked a battery, you really need to try it. Just like Miranda said, you'll never forget doing it. So it creates a current. What happens when the battery's dead? I don't know. I've never licked a dead battery. But what a dead battery is when there's no longer a driving force for the negative charges to flow to the other side. So our membranes are like this, where instead of having the two parts of the battery be separated by some boundary right here, we have sorry, my kindergarten writing here, drawing. Rather than have this kind of separation. We have a separation of the membrane. Positive charges on one side, negative charges on the other side. We have to keep this, we keep a high concentration of positive charges here, high concentration of negative charges here. And if we keep it that way, we have a potential energy source that we can use. If we let things flow via a channel. You allow things to flow down this channel, all the way down. But rather than let the battery just go dead, we have to set it up so that it's a recharging battery. So we're constantly regenerating the separation in charge. So the potential energy source here in our cells is the separation in charge. We can use it to allow for current. Once we allow current to flow, we can use that to send a nerve signal down an axon. So as long as the membrane has a potential or a separation in charge, potential or voltage is a separation in charge. Electrical potential or voltage is a separation in charge. Voltage. Actually, let's do this one. Or voltage is a separation of charge between the inside and the outside of the battery. Inside and outside of battery. A membrane. Cell plasma membrane. A current is a flow of that charge, down the membrane, down the gradient, sorry, flow of charge down the gradient. So as long as there's a separation of charge, the membrane is polarized. It's always polar. Remember, we talked about what polarity meant. So it's separation in charge across a molecule. So this is when there's a separation in charge across the membrane, means that it's polarized. The currents, the charge flow is created by the flow of sodium and potassium through gated channels, which can be opened and closed in response to different stimuli. Currents can be turned off or on. I think, I'm amazed how many people are not here today, and I don't know why. But I think I'm going to give everyone who's here some extra credit because you're here.
Thank you.
You're welcome. Give me 1 second. I'm going to do that right now.
Yay!
I know. Tell me about it. Give me 1 moment. I'm gonna do something really quick. I'm just gonna write down whoever's here. Who's the iPhone? Is that Ashley?
Michaela.
Michaela, I'm sorry.
No, it's fine. I can't fix my name on here, so.
And I know we had more people here, and I know that some of that is related to the fact that people have internet connections that don't work all the time, which I understand. But you do know, you know what? Yeah, I think I will give it as extra credit, but you do know that attendance is mandatory. So I should maybe start docking people for not being here. But I'm not going to do it today. Give me 1 second here. Just here. Catherine. Miranda. Yogesh, Rachel Yogesh. Make sure I have everybody. Here. Okay. I really do want to get caught up, so we will on Thursday we will finish talking about the appendicular skeleton, and then I'm going to do a little bit more lecture. But let's continue forward here. So electrical potentials and currents. The charge difference between the membrane is called the resting membrane potential. So we're constantly resetting this membrane potential, which is a separation in charge. This constant separation charge is a potential energy source. So as long as the battery or the membrane, it has a potential, is polarized, which means that we can use it to do work. concentration of positive charges is higher than the concentration of negative charges on the inside. These potentials are generated because of the ionic gradients. And the fact that the membrane is only selectively permeable to some ions. Sodium and potassium can't move across the membrane without specific channels. Other things cannot pass through the membrane at all. Another reason for Another reason for this potential and the drive to move down the gradient is the negative and the positive charges are attracted to one another. Typically, the inside of the battery, the inside of the cell or the inside of the battery, the inside of the cell is typically about negative 70 millivolts in a neuron. So there's more negatively charged particles inside the cell's membrane. More sodium outside, more potassium inside, and large proteins inside that are negatively charged that can't escape the cell. So this, if you think back on the battery, your typical Duracell battery like this is a 9-volt battery, right? If you look at a battery, 9 volts. The membrane, the electrical potential across a plasma membrane of the neuron is 70 millivolts. So it's smaller, but it's still there. We don't need as much as a 9-volt battery for one cell. So 70 millivolts is the voltage. It's the separation in charge. So measure of the separation of charge, 9 volts in my 9-volt battery, 70 millivolts across the cell membrane of the neuron. So what determines this value? There's leakage channels that will allow more potassium to leak out of the cell and less sodium to leak in. So more potassium can leave the cell, less sodium can come in. There's negatively charged proteins in the cell that can never leave the cell. There's a small inward sodium leak and a small outward potassium leak that is offset by the sodium potassium pump. Remember this one that we talked about when we talked about plasma membranes? It's constantly pumping sodium out against its concentration gradient and potassium in against its concentration gradient. These currents that can be allowed to flow move the ions down the gradients, move them via channels down their gradients. The potassium ions have the greatest influence on the resting membrane potential. And the plasma membrane is most permeable to potassium than any other ion. Potassium can move down its gradient easier than sodium can move down its gradient. So the potassium diffuses freely through ion channels, leaving largely negatively charged ions inside the cell. Stuck inside the cell. However, some potassium can be attracted back by the negatively charged anions inside the cell. The sodium is 12 times more concentrated in the extracellular fluid than the intracellular fluid. So the key player involved in maintaining this resting membrane potential and keeping it reset is the potassium ion. This one thing that we do have to keep in mind all the time right now is that this separation in charge, this attraction between the positive and the negative ions across the membrane, only has to do with right next to the membrane. It's not throughout the rest of the cell. It's just next to the membrane. So the whole entirety of the cell is not negatively charged with regards to the outside. It's just the membrane part right next to the membrane. What if the plasma membrane of a neuron had more sodium leak channels than potassium leak channels? What effect would this have on the resting membrane potential? Where is sodium more concentrated, inside or outside of the cell? Tarek, what would you say?
Would it be outside the cell?
It is outside of the cell, you're right. So if we have more leak channels, meaning and just allowing things to flow, what's going to happen to the negative charge inside the cell? If we have this battery here, negative on the inside, positive on the outside, rather than have it flow that way, the way I have it drawn there. if we allow all the sodium to move to the other side, or even until we lose our concentration gradient of it. We're going to get a dead battery. Is essentially what we end up with, is a dead battery. Once it's equal on both sides, there's no reason for it to keep moving. And so the resting membrane potential would go away. And there would be no potential energy source, no potential driving force. There's 3 kinds of gated channels. We have channels that open. So these are channels which things can flow through if we open or close, if we open them. They can open in response to a chemical stimulus, so a ligand that binds to the channel and opens it, or a ligand that closes it, that binds to the channel and closes it. We can have channels that are gated that open in response to some sort of stimulus, mechanical stimulus, such as calcium binding. We can have some channels that will change and will respond, open and close based on a change in the electrical potential, membrane potential. So chemical, mechanical, and electrical. So chemical, specifically ligand-gated channels, open and close in response to specific channels, excuse me, specific chemicals. These ligands or chemicals can include neurotransmitters, hormones, or ions. We find these highly concentrated at neural synapses. There are numerous in dendrites and cell bodies of interneurons and motor neurons. Mechanically gated channels open and close in response to mechanical pressure or vibration. They're found in hearing receptors, so they kind of get bumped and then they fire. They open in response to the fact that they got bumped. An example of this is applying a pressure to a receptor. They get stretched and they open. So, most often it's a pressure change. They get nudged and they open. Voltage-gated channels open in response to a change in the membrane potential. The membrane potential goes from being -70 to -50. And that opens up the channels. These type of channels are specifically found along the neuronal axon. And they participate in the generation and conduction of action potentials, which we'll start talking about today. We'll talk more about it next time. So they're mainly located along the neuronal axon. Neurons can communicate via 2 different types of potentials or electrical signals. Potentials are the same thing as electrical signals. Electrical potentials. changes in electrical potential or membrane potential. So graded potentials are work only for short distance communication. Action potentials communicate over long distances. For example, you touch a pen, the graded potential develops in the sensory receptor in the skin. Once the inner neuron, remember that this is the same thing as what we saw, sensory neurons, inner neuron, motor neuron. Once the inner neuron in the cerebral cortex is activated, perception occurs. So you can feel the surface of the pen. Graded or local potentials are changes in the respiratory potential, membrane potential of a neuron, occurring at or nearby the part of the cell that's stimulated. These local graded potentials, so local potential is the same thing as a graded potential. Local potential is a graded potential. So local potentials are graded, meaning they vary in magnitude according to the strength of the stimulus. The more intense and prolonged a stimulus, the more gated ion channels can open for a longer time. The result is more sodium can enter the cell. The voltage changes more than it would with a weaker stimulus. So it can vary in magnitude, and they can become bigger with a stronger and more prolonged stimulus. But at the same time, they're decremental, which means that they get weaker as they spread from where they originated. That meaning that they're only used for short distances because they diminish with distance. They're reversible. Once the stimulus stops, the diffusion stops. And the membrane will quickly return back to the original resting membrane potential. They can be excitatory or inhibitory, which means that they can hyperpolarize, make the membrane more negative, inside of the membrane more negative, or make it more positive by depolarizing it. To depolarize the membrane means that it makes it more, that negative 70 millivolts, more positive. If you hyperpolarize the membrane, you make it more negative. The negative side, but you make it maybe negative 75 millivolts. Acetylcholine is a neurotransmitter that usually causes depolarization, causing the membrane potential to become more positive. So the inside becomes like maybe negative 65 millivolts. Glycine causes it to become more hyperpolarized. The cell's membrane becomes more negative. So maybe rather than negative 70, it's now negative 75 millivolts. When it becomes more negative, it's more likely to reach the threshold level and fire an action potential, which we'll talk about next time, but I want to finish talking about graded potentials here. There's a balance between these two types of local potentials. So graded means that the membrane potential can vary in amplitude. The greater the stimulus, the larger the amplitude. Mom, can you wait just a minute? This relationship between the stimulus strength and the amplitude also exists in a hyperpolarizing graded potential. Graded potentials can also sum up, but they die out with decremental conduction. They can add together, where one graded potential adds on the effects of the last graded potential. Or they can die out with time. A depolarizing graded potential, the stimulus causes a reduction in the difference charge. Hyperpolarizing, the cell becomes more negatively charged. Most graded potentials occur in the dendrites and cell bodies. They do not travel down the axon. They're going to occur up here. Questions? Graded potentials are going to occur as a result of opening or mechanical ligand-gated channels. And the membrane potential will become more hyperpolarized or depolarized depending on the type of channel that opens. Which type of potential does not decay with distance? I don't think you're going to be able to answer this yet, and I probably should put this somewhere else, this question. Action potentials are not going to degrade. They're an all or nothing. Actually, I think we're going to stop right here. Are there any questions?
No.
Okay. Let's stop right here. And I will see you on Thursday. We will finish up the appendicular skeleton. We'll actually probably go back to the beginning, and then I want to do some more lecture. Have a wonderful 2 days.
You too. Thank you.
You too.
Thank you.
Thank you.
Miranda and Michaela, are you here?
Yeah, I'm just trying to think of if I have any questions.
Okay, that's all right. I understand.
I will ask you to open up 8 and 9 for me. I emailed you last week, but I did not get the email back.
I thought I emailed you back. I think I did that. Let me, bear with me. I'm gonna stop recording just